No.
So, good morning, good afternoon, good night. For those that are going to go to bed, don't worry, you can listen to it and wake up. My name is Antonio Baines. I'm an associate professor at North Carolina Central University in Durham, North Carolina. I'm a cancer biologist, and I specifically study pancreatic cancer. And today I would like to talk to you, or tonight, or depending on what time zone you're in. I want to talk to you about the research I've been doing over the past years as it relates to pancreatic cancer and oncoproteins K-Ras and Pim kinases. So let's get started. So I had a video, but we're not going to worry about that. But I had a video to put a face to pancreatic cancer, and the person I had was basically was an actor who came down with pancreatic cancer and died, Patrick Swayze. And so again, pancreatic cancer is a very deadly disease, and it can affect anybody unaware. And so again, there's much research that needs to be done. So here's a picture of the pancreas, just to refresh your memory from your anatomy class. The pancreas is an organ in the gastrointestinal tract. It has two major functions. It makes digestive enzymes that get secreted from the pancreas into this pancreatic duct into the small intestine to aid in digestion. Another function of the pancreas, it makes hormones, hormone like insulin, to control blood sugar. Okay? And so if the pancreas is damaged, we have problems. So in 2012, the American Cancer Society ranked pancreatic cancer as the fourth most common cause of cancer deaths in the U.S. And you can see here it affects both men and women. Now, the problem with pancreatic cancer, although it may not be as high as lung cancer, for example, the problem with pancreatic cancer is that, unfortunately, it kills most of its patients. And so in 2012, there was about a 75% mortality rate as it related to pancreatic cancer. And I believe in 2013, this year, that rate has actually gone up to around 80–85%. So again, a very lethal cancer. Now, some risk factors to consider when we talk about pancreatic cancer include some of the following. Smoking is a really huge risk factor as it relates to pancreatic cancer, as well as diabetes, long-standing diabetes, chronic pancreatitis, and various hereditary diseases such as hereditary pancreatitis. Okay. Now, in terms of challenges of pancreatic cancer, there's quite a few. There's quite a few that lends the need for help in the field as it relates to trying to better tackle pancreatic cancer. One is usually diagnosed at an advanced stage. It usually is already metastatic, is a metastatic disease when it's usually found, and it usually is found in various organs such as the liver and lungs. Another challenge is that there's a small percentage of people who are eligible for surgery when diagnosed with pancreatic cancer, but it's usually less than 15% of those. 15% of that population are eligible for surgical resection, and even with the surgical resection, it still doesn't guarantee a long life, unfortunately. Another challenge is chemotherapy. Most pancreatic cancers are resistant to chemotherapy or become resistant over time to chemotherapy. And unfortunately, chemotherapy really only prolongs one's life around half a year. So again, definitely not much is done with chemotherapy as it relates to survival. And even with some of the recent findings in terms of combinations, doing combination therapy, it still really doesn't prolong one's life significantly. So again, at the end of the day, there is definitely a need for more research to help combat this fatal disease. So pancreatic cancer, just as with many other cancers, it takes time for that cancer to form. And the reason why there's this timeline is because it takes time for normal cells to acquire various mutations, even oncogenes like K-Ras, or various tumor suppressor genes like p53. It takes time for mutations to occur in those different genes to promote the progression of normal cells to becoming more and more aberrant and eventually becoming cancerous. And so again, this slide here is just showing some of those players that are involved in that process of normal cells becoming cancerous in the pancreas. So again, looking at statistics that American Cancer Society came out with in 2012, this shows on your left-hand side, this left column shows the variety of cancers and the percentages in terms of cancer deaths that occurred in 2012. And you see the highlighted ones I have, they're lung, colon, and pancreas. So they make up the top four. They're in the top four cancers that unfortunately most people succumb to. And if you look on your right-hand side to the column I have tumor types as well highlighted, some cancers highlighted. Again, pancreatic, colon, and lung cancers, which again make up some of the cancers we saw on the left-hand side. And in this case, we're looking at Ras mutations. We're looking at the percentage of Ras mutations found in those cancers. And if you notice, pancreatic cancer, we have 90% of all pancreatic cancers have Ras mutations. 50% of all colon cancers have Ras mutations, and about 30% of lung cancers have Ras mutations. So if you notice the top three cancers, out of the top four cancers we have, 3 of them have significant percentages of Ras mutations, which suggests Ras may be important for those cancers. Now, this may be reviewed for some and maybe new for others, but just to make sure we're all on the same page, Ras is an oncogene. In addition to it being an oncogene, it's a GTPase, and so it's an enzyme that catalyzes GTP to GDP. So it catalyzes GTP to GDP, and when Ras is bound to GTP, it's on in the normal cell. It's on in the normal cell, but it's off when it's bound to GTP. And so when Ras is activated due to some extracellular stimuli, when Ras is activated, Ras bound to GTP will signal down various effector pathways. That's what the E stands for. The yellow circle with the E stands for downstream effector pathways that Ras can signal through to cause various phenotypes in the cell. So, in the normal cell, Ras turns on and off. So again, bound to GTP is on, but when the upstream signal stops, Ras will catalyze GTP to GDP and turn off. Okay. Now, in the cancer cell, in figure B, in the cancer cell, when Ras is mutated, and that's what the asterisk in the blue circle Ras, the asterisk is referring to a mutation. When Ras is mutated, Ras is bound to GTP and cannot catalyze GTP. So Ras is bound to GTP and is constitutively activated, and it leads to persistent deregulated signaling downstream of it, down various effector pathways. Okay, so again, when Ras is mutated, Ras is not able to catalyze GTP to GDP, and it leads to persistent downstream signaling. And that's what usually can lead to various phenotypes like excessive growth that can lead to cancer. Now, this slide is showing various downstream effector pathways of Ras. Okay, so we have the RAF, some of the common pathways of Ras include the RAF-MEK-ERK pathway in the middle, and then we have the PI3-AKT pathway, the RalGTP pathway, and some other pathways. So again, these are various downstream effector pathways that Ras can signal through to lead to various phenotypes in the cell. And again, the asterisk is referring to what's mutated in cancer. So again, Ras can be mutated as well as some of the various players in some of these pathways downstream of Ras as well. Oh, and I forgot to mention, I will be taking questions, so feel free to ask questions, but I will verbally answer them at the end of the presentation. I apologize, I supposed to have stated that earlier. So let's continue. So again, just providing more background about Ras. So there's different types of Ras, and these are some of the major types of Ras. There's HRAS, NRAS, and K-Ras. There's two different isoforms, 4A and 4B. Okay, so so that's one piece of information about Ras. But in the past, traditionally, HRAS has been the focus due to reagent availability. Okay. So studies have shown from various investigators that 32% of all human cancers have Ras missense mutations. Okay, so Ras is it's found very prevalent in various cancers. And again, Ras, there's different types of Ras, and this is showing the components that make up that percentage, that 30% percentage. We have K-Ras 21%, and NRAS and HRAS lesser amounts. Okay. And again, various studies have been done. A lot of cancer tissues and cell lines have been sequenced to really get at the question, well, what Ras isoform should we be focusing on? What's the main Ras mutation that we see in cancer? And various entities such as the Consortium of Somatic Mutations in Cancer, COSMIC, and other entities have found that about 85% of Ras mutations that we see in cancer, 85% of them are K-Ras mutations. And then we see NRAS and HRAS. So again, although HRAS was traditionally focused on in the past, recent sequencing, gene sequencing studies, sequencing cancer tissues and cell lines have shown that K-Ras seems to be the major Ras that's mutated in cancer. And again, as I mentioned earlier in the previous slide, again, almost all pancreatic cancers, half of colon cancers, about a third of lung cancers, and a quarter of melanoma cancers all have various Ras mutations. Okay, and so basically from these studies, it is very clear that K-Ras seems to be a major player as it relates to being an oncogene, being one of the major oncogenes mutated in cancer, as well as being, as it relates to the different types of Ras mutations, being the major Ras that is mutated in cancers. And so this leads me to the major goals that I've focused on as a postdoc and as a faculty member as it relates to Ras and pancreatic cancer. So first research goal, basically to determine the requirements for and the function of K-Ras in pancreatic cancer. Okay, so for example, let's ask the question, is just because K-Ras is mutated in many pancreatic cancers, does that mean it's really needed for the growth, progression, development, maintenance of the cancer? So again, it gets at the question: is K-Ras a driver, or a passenger mutation? Two, another research goal is validate novel molecular drug targets downstream of K-Ras in pancreatic cancer. Again, the idea is, okay, again, not only is Ras a concern, but who Ras talks to can be an issue and maybe a possible target for therapeutics. And then lastly, understand again the role of these molecular targets, Ras and downstream targets of Ras, as it relates again to the development, progression, maintenance of cancers in the pancreas. So those have been kind of the overall goals I've focused on over time. So for this presentation, my data will hopefully be focused mainly on the first two research goals that I just mentioned. So again, is K-Ras needed for pancreatic cancer growth and development? And so to get at that question, what we did, we took a pancreatic cancer cell line, I think specifically CAPM1, which has a K-Ras mutation, K12B mutation, and we used shRNA and knocked down K-Ras, the mutated K-Ras. We suppressed transcription, which prevented translation, which led to this Western blot that shows very small amount of K-Ras protein compared to the control cells, the vector cells. And so we're showing here that we were successful in significantly decreasing mutated K-Ras in this mutated K-Ras cell line. And to kind of confirm that this shRNA worked, we looked at a downstream pathway of K-Ras, the raffinate ERK pathway, specifically looked at phosphorylation of ERK, and we saw in the cells that we were able to decrease mutated K-Ras, we saw also decreased phosphorylation of ERK as well. Okay, so again, wanted to show that we can modulate K-Ras, mutated K-Ras expression. And then we wanted to look to see what role, did mutated K-Ras play in certain types of growth. And so one of the assays that we used in the lab was the soft Auger assay, which measures anchorage-independent growth. And so normal cells, so basically you have a plate with cells, normal. So if you have a plate with auger in it, and if you put normal cells in that auger, they won't grow because they need to be anchored to something to grow. But tumor cells, cancer cells, they don't care. And so you put tumor cells in that plate with auger, they just grow and form colonies, and they're just perfectly happy. And so one of the questions we wanted to ask was, if you inhibit mutated K-Ras, will you prevent this type of growth from occurring? And the importance of this particular assay is that this assay relatively predicts what happens in xenograft animal models. Okay, so what usually happens in the assay usually happens if you put those cells in the xenograft model. So again, we use this assay to look at anchorage-independent growth. And so here you're looking at data where we took those same cells that I showed you earlier, vector cells, basically the mock shRNA that doesn't inhibit anything, and then cells where we knock down and significantly decrease mutated K-Ras expression. And as you see, between the vector cell and knockdown cells, we see significantly decreased size of the colonies in knockdown cells, suggesting that mutated K-Ras is needed for that type of growth, because when you inhibit K-Ras, you see definitely significant decrease in colony size. Okay. And then we also took those cells, and again, as I said before, this assay is important because it helps to predict what potentially would happen in an animal model. And we showed that, so here you're looking at a graph, days on the x-axis, and on the y-axis we have tumor volume in cubic centimeters. And as you see, we have vector cells versus the cells, knockdown K-Ras. And as you can see, when we significantly decrease K-Ras expression, we also significantly decrease tumorigenicity of the cell line in the xenograft model. So again, showing that mutated K-Ras is needed for growth, anchorage-independent growth, as well as tumorigenicity. And what makes this data very interesting is that, again, going back to the question I asked before, okay, we know K-Ras mutations occur early on in the progression of normal cells becoming aberrant and potentially leading to cancer. And so, but the question becomes, do we need mutated K-Ras all throughout that process? And so these kappa-1 pancreatic cancer cells are adenocarcinoma cells, so they are advanced cancer cells, and we show that when we inhibit mutated K-Ras, we still can impact the growth of that cancer, suggesting that mutated K-Ras is still important even after the cancer has advanced to a late stage and formed an adenocarcinoma. Okay. So again, just wanted to remind you that, again, K-Ras, K-Ras signal down various signaling pathways to produce various phenotypes, such as excessive cell growth, or if it's mutated. And so again, some of the common pathways that scientists study, and I've looked at as well. Again, the raffinate ERK pathway, the PI3AKT pathway, and again, I've studied both of those pathways as it relates to Ras, but due to time, I won't be able to show that data today. But what I will do is focus on this third pathway that you may or may not be familiar with, called the RalGTP pathway. Okay, so what is this RalGTP pathway? Well, that pathway involves RalGTPases, so which is another member of the Ras branch of the Ras superfamily. So these RalGTPases, which are activated by RalGTPs, are the two members are RalA and RalB. Okay, again, GTPase is just like Ras, and so they catalyze GTP to GDP. Just some basic information about these proteins. So again, we have RalA and RalB. They're very similar to each other. They share a very high homology as it relates to amino acid identity. And again, they are involved in potentially cancer growth as well, being downstream of the Ras. So here you're looking at some data. So here we did a GST pull-down assay, and we used a variety of different pancreatic cancer cells. We used a variety of different pancreatic cancer cells that had K-Ras mutations. And so again, the GTS pull-down assay, the pull-down activated RalA. So again, RalA down to GTP is activated. So we pull that down using the GST pull-down beads. And as you can see, there is high levels of RalA-GTP in the majority of these different pancreatic cancer cell lines. And we also look at total RAL protein as well, just for comparison. And again, although I'm not showing you the data that we did for looking at phosphorylation ERK and phosphorylation AKT in pancreatic cancer cells, we look at those levels in these cell lines as well. Although I'm not showing you the data, what we did see some activation, but it definitely was not as frequent as we see with activated RalA. So again, definitely saw activation of ERK and activation of AKT in these different pancreatic cancer cells, but we definitely didn't see it as frequent as we do see with activation of RalA. We also looked at RalB in the pancreatic cancer cells, and if you notice, again, so we looked at RalB, and if you notice, we do not see the same level of frequency of activation of RalB as we saw at RalA. So the bolded arrows shows you the cell lines that had the high amounts of activated RalB, the most amounts of RalB. So this suggests that although RalA and RalB are very similar in terms of amino acid identity, there apparently are differences because we're seeing definitely differences in activation of RalA and RalB. And studies have shown since this data that there are definitely differences between RalA and RalB in terms of function. So again, just to recap again, we looking at a downstream effective pathway of Ras, specifically the RalGTPase pathway. And again, the idea is Ras is mutated in almost all pancreatic cancers, but the question becomes, what pathway or pathways is Ras signaling through, is K-Ras signaling through to lead to this aberrant phenotype of tumorogenicity, for example. And so that's just want to recap and why we're looking at this. And so so I just showed you that RalA seems to be activated more so than RalB in a variety of pancreatic cancer cell lines. And so the next question becomes, well, what's the role of RalA and RalB? Especially as it relates to like cancer growth, for example. And so what we did, just as we did before, we used shRNA to knock down, in this case, either RalA or RalB. So we either knock down RalA and RalB. So in yellow is the RalA knockdowns in these 3 different pancreatic cancer cell lines that you see above here. And then in the white, you see the RalB knockdowns. So again, we made shRNAs against either RalA and RalB, and using lentivirus, infected the cells with these viruses that contain these shRNAs for stable expression of the knockdown. And the vector cells, again, are basically mock shRNAs. Okay, and so first of all, we're looking at a Western blot. So we're looking at Western blot just to confirm the knockdown was specific, because again, since RalA and RalB seem to be very similar, we want to make sure that our knockdown was specific. And so in the first row of Western blots. We confirm RalA knockdown with the RalA knockdown, but we don't see any effects on RalB, which is good, showing specificity with RalA knockdown. And then the second row with RalB, we show that we knock down RalB successfully, but not RalA. Okay, so again, just showing that our knockdowns, our shRNAs, seem to be selective and not affecting the other Ral protein. And then what we did, we took those cells, either RalA and RalB, and put them again in the soft log assay. And if you remember again, I mentioned that assay helps to predict what happens in a xenograft animal model. And so we put all those cells in soft log assay to see if it would impact, see if knocking down RalA or RalB would inhibit soft organ growth, anchorage-independent growth. And if you notice, inhibiting RalA definitely significantly decreases the number of colonies compared to the mock vector cells, but we don't see really any significant decrease when we knock down RalB. Again, suggesting that, again, RalA and RalB are playing distinct roles as it relates to pancreatic cancer, and it seems that RalA seems to be more important for anchorage-independent growth in pancreatic cancer. And what we did then is, again, went to the animal model, which is the xenograft animal model, and we showed that my colleague was able to show that, again, we took these same cells that, and these are the same cells, if you remember, these are the same cells that had mutated K-Ras as well. So we took these, these cells, knocked down RalA and RalB in these cells, and implanted them into a xenograft model and to look at tumorigenicity. So the graph on the left, again, the x-axis is time, days, and the y-axis is volume. And again, we have scrambled shRNA, which is the shRNA, the control shRNA, and we have knockdown, we have RalA shRNA, RalB shRNA, and then we have a rescue as well of the RalA. So as you can notice, when we knock down RalA, we see a significant decrease in tumorigenicity. But when we knock down RalB, we don't see really any decrease in tumorigenicity. Okay? And the picture on the right basically corresponds with the graph on the left. Now, to confirm that this RalA knockdown was really specific, our colleague did a rescue where he added back in RalA and basically was able to rescue those tumors. We were able to basically rescue those tumors. So again, showing the picture here, again, when you knock down RalA, you get definitely a significant decrease in tumorigenicity, but then when added RalA back to the equation, was able to rescue the tumor growth, so just suggesting that again, RalA is necessary for CAPN1 tumorigenic growth. But as as we all are interested in seeing what happens in the patient, we took this to the patient system, and what we did, what our colleagues did, they were able to get tissues, either normal pancreas tissues or tumor tissue. So the N stands for normal pancreas tissue, and the T stands for tumor tissue. If you notice, you also see matched and unmatched. So the matched pairs, normal and tumor, refers to the tissues coming from the same organ, the normal, area of the organ, as well as the tumor part of the organ of the pancreas. And the unmatched is referring to samples came from different locations, different pancreatic, different pancreases, either tumor tissue of the pancreas or normal pancreas, but basically from different sources. And as you can see here, again, did a pull-down assay to pull down activated RalA and RalB, and as you can notice, we see that there's definitely more activation of RalA and RalB in the tumor versus the normal. And so you can see that definitely the RalA-GTP and the RalB-GTP. Okay, and what's more interesting, what's just as interesting is if we go down and look at phospho-AKT, phospho-ERK, you don't see much activation of AKT. You don't see much phosphorylation of AKT or phosphorylation of ERK compared to the activation of RalA and RalB. So again, providing more evidence that again, not saying that those other pathways are not playing a role, but we're definitely seeing more activation with, we definitely see more frequent activation with these Ral proteins. So summary of the results. So one, hopefully I've been able to show you that pancreatic cancer cells seem to depend on continued expression of mutated K-Ras for transformation in vitro and tumorigenicity in vivo. So again, it's not just, yes, although K-Ras mutation is found early, it seems to still be playing a role even throughout the progression of normal cells becoming more aberrant and eventually become cancerous. Also, hopefully shown you that of the three major effective pathways, the RAL pathway seems to be frequently activated. And between RalA and RalB, it seems that RalA seems to be very important in activation. The activation of RalA seems to be very important in the tumorigenicity and oncogenesis of pancreatic cancer, more so definitely than RalB. So Ras has been studied for many years. I think it's been about 30, maybe close to 40 years now. Ras has been studied, and again, because Ras is mutated in so many cancers, and as I showed you earlier, it's definitely a player in. It definitely seems to be a player in 3 of the major cancers, of the top 4 cancers that people succumb to: pancreatic, colon, and also lung cancers. People have believed that, scientists have believed that Ras would be a great molecular target. And so many researchers in academia, industry, government have done tons of research on trying to find ways to inactivate or inhibit Ras to, again, have an impact on cancer. So a variety of things have been done. So it shows just a kind of a quick cartoon. So again, Ras has to go to membranes. Usually Ras has to go, gets to transport the membranes to get taken to the membranes to be activated. So people try to inhibit that process. People try to inhibit effective pathways. People use viruses and all types of techniques to try to inhibit Ras. But unfortunately, to this day, no anti-Ras drug has been developed in the clinic. Has successfully made it through the clinic and is being used on patients for a variety of reasons, And so again, the question becomes, how can we target Ras? How can we target Ras to potentially have an impact on cancer? And so one of the ideas that have come out from the many years of research on Ras is, well, if we can't target Ras directly, maybe we can target Ras indirectly through potentially modulating who Ras talks to. So Ras, as you saw, there's a variety of downstream effective pathways that Ras signal through to cause various phenotypes in the cell. And so the question becomes, maybe we can have an impact on gene expression based on who Ras talks to. Okay, and so that leads me to the next topic that I want to cover in my presentation, which is the target that I study currently in my laboratory now. So a paper came out in 2005 where basically the investigators took a normal pancreatic ductal epithelial cell, transformed it, immortalized it, and overexpressed mutated K-Ras in that cell to make it tumorigenic, and they did microarray analysis on it. With the idea they wanted to see, what genes would be upregulated and downregulated in those cells. And one of the genes that came out of that microarray analysis was a member of the PIM kinase family called PIM1. So, what are PIM kinases? So first of all, the family of PIM kinases involve 3 members, PIM1, 2, and 3. And PIM stands for the proviral integration site for the Maloney-Murine leukemia virus. Okay, so it's first found in lymphomas and leukemias, but now people have found it to be involved in various solid tumors. So this is showing, again, 3 members of the PIM kinase family. They're specifically serine threonine kinases. They're very similar to each other. So PIM1, as it relates to PIM1, so PIM1 and PIM2. PIM1, PIM2, PIM3. Okay. Some information about these specific kinases. They are very unique in that they lack a regulatory domain, and they are also believed to be constitutively active. So when you see them expressed, it is believed that they are constitutively active when you see them expressed. They are believed to not be regulated by phosphorylation or membrane relocation, as many other kinases are, and they are regulated at the transcriptional level. They can be regulated by protein stability, and they can also be regulated by proteasome degradation. So just some more information about PIM kinases. So PIM kinases, they're oncogenes, they're anti-apoptotic. PIMs are downstream of the JAK-STAT pathway. They're downstream of the JAK-STAT pathway. And PIMs, again, since they're kinases, phosphorylate a variety of substrates to either turn them on or off, which can result in various phenotypes in the cell, as you can see here. And also you can see that in this cartoon as well. So again, PIM kinases play a role in a variety of different processes in the cell, as you can see here. And again, they do that by phosphorylating a variety of substrates. And one of the main, so again, as you can see, there's a variety of functions in the cell that PIM kinases seem to be involved in, but we focus a lot on cell survival and apoptosis. So I became interested in PIM kinases in pancreatic cancer, one from the paper I just showed you earlier where they did a microarray study, but also this paper here that came out in 2006, where they were able to show that PIM3, one of the members of the PIM kinase family, was aberrantly expressed in pancreatic cancer and was found to be involved in apoptosis in those pancreatic cancer cells. So this paper kind of gave me increased my interest in possibly looking at PIMs in pancreatic cancer because, again, as I mentioned earlier, PIMs were mainly focused on in hematopoietic cancers like leukemias and lymphomas. But again, studies start showing that they play a role also in solid tumors, especially prostate cancer. And then this paper came out showing that it was also believed to be involved in pancreatic cancer as well. So came up with 2 hypotheses as it relates to PIM kinases and pancreatic cancer. So the first hypothesis was kind of getting at the question of the role that RAS may play in PIM expression. And again, remember that previous paper I mentioned about the microarray study. They showed that one of the many genes that were upregulated when K-Ras was overexpressed in that normal pancreatic ductal cell was PIM1 kinase. So I want to, get at, want to further investigate that further. Further investigate that. And then 2, what happens? What's the role of PIM kinases in pancreatic cancer? If you inhibit PIM, does it play a role in growth of the cancer? So again, 2 hypotheses that we investigated. So here we show that. So again, the idea of if we can't target RAS directly, maybe we can target RAS indirectly through some of the players that K-Ras talks to. And so, one we want to know, okay, well, maybe PIM is some target that we should focus on. So first, we wanted to show that it was that there was more PIM in tumor tissues than the normal, more PIM1 in the tumor tissue than the normal. So here we're looking at Western blots. In A, we're looking at Western blots, and again, N stands for normal, T stands for tumor. Those numbers that you see are referring to densitometry readings that we did to show significance between the expression between normal and tumor as it relates to PIM1. And as you can see, there's more in the tumor. There was more found in the tumor tissues than the normal tissues as it relates to PIM1. And then we decided to look at cell lines, look at a variety of different pancreatic cancer cell lines, and compare them to a normal pancreatic ductal cell HPNEs right here, HPNEs in B. And as you can see, there's much more PIM1 expression, protein expression in the cancer cell line versus the normal cell line. So again, showing that PIM1 protein is upregulated in human pancreatic cancer tissues and cell lines. Here we wanted to get at the question of the role K-Ras plays as it relates to PIM1 and the other PIMs as well. And so let's start with figure A. So in figure A, so just to be, so at the end of the day, again, we're looking at Western blots. So just to give you some orientation, so on the left-hand side, we're looking at the different types of different proteins, K-Ras, the 3 different PIMs, and of course beta-actin for loading control. And then the first column is HPNE cells, so those are normal pancreatic ductal epithelial cells. And then the second column is HPNE K12D cells, so those are cells that were transformed with mutated K-Ras. And so, as you can see, and as you would expect, you see more K-Ras protein expression in the HPNE K-Ras 12D cells versus HPNE cells. Okay, that makes sense. But then we want to look at the various PIM expression, protein expressions, and as you can see, we see an increase in PIM1 and in PIM2 as it relates to protein expression in those transformed HPNE cells. When we increase, so basically when you increase K-Ras, we get an increase in PIM1 and PIM2, but not PIM3. Okay, in figure B, we wanted to get at the question, okay, well, what happens when you knock down K-Ras? What happens when you decrease K-Ras? And so we took 2 pancreatic cancer cell lines, Neopockets and Kaplan-1 cells, and we we infected these cells with shRNA against mutated K-Ras to give us knockdowns. So we have decreased mutated K-Ras in Neopockets and Kaplan-1s. And as you can see, we definitely, we see a decrease in, in K-Ras expression, especially in Kaplan-1 cells. And then we decided to look at PIM1, PIM2, and PIM3 in those cells. And really the only PIM that we saw an effect on as it relates to a decrease was PIM1. We didn't see any changes in protein expression of PIM2 and PIM3 when we knocked down K-Ras. But with PIM1, as I have highlighted, we saw a decrease in PIM1, suggesting that PIM1 may be the major PIM that's modulated by modulated by K-Ras, because again, when we increase K-Ras, as we have mutated K-Ras, as we have in figure A, we see effects on PIM1. We saw effects on PIM2 as well, but PIM1. But when we knock down or decrease K-Ras, we see only effects on PIM1. So suggesting that PIM1 may be the main PIM isoform to be modulated by K-Ras. Okay. And then, again, we'll get the question of the function of PIM kinases in pancreatic cancer. And so we, again, used shRNA technology and made shRNAs against PIM1. Of course, we use different shRNAs because we never know which one can work the best. And as you can see, so again, both cell lines, Neopockets and Kaplan-1s, we were able to have various levels of knockdown against PIM1 in the cells, and to confirm this knockdown, we saw, we also looked at one of the main downstream targets of PIM kinases, BAD, the proapoptotic protein BAD, phosphorylation BAD. And so, as you see, when we knock down, in cases where we have greater knockdown of the PIM1, we saw corresponding decreases in the phosphorylation of BAD as well as we saw also effects on total BAD expression as well. So basically, after making those cells, we now have a tool to do studies to investigate what's the role of PIMs on different phenotypes in the cell. So one, we decided to do a 6-day growth curve on those cells where we knock down PIM expression. And as you see, we were able to see a significant decrease in, so again, we have days on the x-axis and cell number on the y-axis. And as you see in both figures, Neopockets and Kaplan-1s, in the cells that we had greater decrease of PIM1 due to the knockdown, we saw greater decrease in growth of the pancreatic cancer cells compared to the scrambled shRNA cells that basically similar as communal cells. We also not only look at anchorage-dependent growth, so not only growth on plastic, but again, as you are familiar with, we went back to our soft auger assay to look at anchorage-independent growth as well. Again, just to remind you, this is indicative of what usually happens in xenograft animal models. And so again, took those cells, put in the soft auger assay, and looked at colony growth formation. And as you can see, so we got pictures of that as well as a graph showing that as well, showing that data. Again, we see compared to the scrambled shRNA that when we knock down PIM1, we do have an impact on colony number and size when PIM1 is inhibited compared to the control. And we also looked at the other cell line, and you can see, more significant decrease in colony number compared to the controls when we knock down PIM1 expression, or decrease PIM1 expression. We also looked at invasion. Pancreatic cancer is a very invasive cancer, and so again, so basically, again, looking at both cell lines, neopockets and capital worms. Again, x-axis, different knockdown groups, and y-axis, the number of invaded cells. We used a Neutrogena assay, and as you can see, in both cases, when we inhibit PIMs at various levels, we see a decrease in invasion, suggesting that PIM1 is needed for invasion of pancreatic cancer cells. We also looked at radial resistance, radiosensitivity. Pancreatic cancer is very radial resistant, and so we show at the high dose of radiation, 7 gray, that when you decrease PIM1 expression, you make those cells more sensitive to radiation treatment. So in black is the scrambled. The black bars are the scrambled shRNAs controlled, and the gray are the PIM1 knockdowns. And again, at the highest dose, we definitely see that inhibiting PIM makes the cells up to sensitize, radiosensitize the cells. So I've shown you basically when we inhibit PIM protein expression using shRNA to disrupt transcription and which prevents translation. So I've shown you what happens when we inhibit the expression of PIM, but what happens when you inhibit the activity of PIM? What happens when you inhibit the activity of PIM kinases with using inhibitors? And so I have the privilege of working with various pharmaceutical companies. Pharmaceutical companies are very interested in these PIM kinase inhibitors, or in developing PIM kinase inhibitors. Actually, there's I think 2 companies that have PIM kinase inhibitors in phase 1 clinical trials now. So just to give you an appreciation how important the pharmaceutical industry feels about this particular molecular target. And so, again, I have the fortune of working with various companies, such as this company Supergen that has changed its name to Astex. And so we've done some work with their inhibitor, this SGI-1776, and we show that when we treat cells with different amounts of the PIM inhibitor, and look at phosphorylation of Bad, we see a decrease in phosphorylation of Bad, suggesting that this inhibitor is having an impact on PIM phosphorylating Bad, Bad apoptotic protein. We also show that when we use this inhibitor and treat cells with this inhibitor and look at cell cycle analysis, we see, so we get flow cytometry data, so this is flow cytometry data, and looking at the different phases of the cell cycle, and as you can see, between 0 micromolar and 5 micromolar, we can definitely see that the cells seem to be getting stuck in G0, G1 as compared to the other stages of the cell cycle. So suggesting that this inhibitor causes cell cycle changes in pancreatic cancer. And we also was able to show that when we combine this inhibitor, this PIM inhibitor, with chemotherapy, in this case gemcitabine, which is the main drug used against pancreatic cancer, we're able to show this combination leads to synergistic effect as it relates to cell viability. So again, looking at two different pancreatic cancer cell lines, x-axis drug concentrations, and we have either untreated, or we have cells treated with gemcitabine, that's a GEM, or SGI, the inhibitor, or the combination of the gemcitabine and inhibitor together, PIM inhibitor together. And in both cases, you see a significant decrease in the combination compared to each of the drugs alone, especially in the neopopulace. In neopopulace, we definitely see a significant synergistic decrease in cell viability with the combination compared to the individual drugs alone. And so, in summary of the results, so we show here that PIM1 kinases increase in cancer cell lines and tissues versus normal pancreas. We show that PIM1 kinase seems to be important for pancreatic cancer growth, either anchorage-dependent on plastic or anchorage-independent in the soft Barber assay, which again representative of xenograft models. And then we show that PIM1 kinase is required for invasion and radioresistance. In addition, we show that the PIM inhibitor seems to decrease phosphorylation of the bad protein and induce cell cycle changes. And we also show that the combination of gemcitabine and the inhibitor significantly decreased cell viability. And so the conclusions, one, we believe that PIM1 kinase levels may be a useful molecular marker or target for mutated K-Ras activity in pancreatic cancer cells. So again, the idea that since PIM1 seems to be modulated by K-Ras, it may be a possible marker to help us determine the K-Ras activity in that particular cell line or tissue, as it relates to cancer. Also, I show data that PIM1 seems to be a good modulator of radioresistance in pancreatic cancer, and that inhibition of PIM kinase can modulate chemoresistance in pancreatic cancer. So when we use that combination of the PIM inhibitor and the gemcitabine chemotherapeutic drug, we saw a greater decrease in cell viability, making cells more sensitive to that drug. So future studies. So there's still much work that needs to be done as it relates to PIM kinases. So one interest I have is what's upstream of PIM kinases as it relates to Ras. So we know PIM is downstream of the JAK-STAT pathway, but what's activating PIM kinases that may what is Ras? How is Ras activating PIM kinases? What's in between Ras and PIM kinases, for example? Two, I'm interested in identifying and validating novel downstream targets of PIM kinases in pancreatic cancer. We know quite a few substrates of PIMs, but again, I believe that might vary depending on the cell context, and again, and not only cell context, but in terms of what pathways that PIMs are using to lead to again carcinogenesis, development of cancer. Interested in validating these PIM inhibitors that these companies are making, various companies are making. Interested in identifying validating these inhibitors in various mouse models of pancreatic cancer. And then lastly, I'm interested in testing these inhibitors in combination with other chemotherapeutic drugs. So again, can we find a good combination of chemotherapeutics, and PIM inhibitors that will lead to making the cells more sensitive against various treatments. Again, this work would not have been possible without a variety of collaborators, not only my laboratory at North Carolina Central University, but collaborators at UNC Chapel Hill, Duke University, University of Utah, as well as various pharmaceutical companies such as Astex Pharmaceuticals. Definitely a team effort. And just want to remind you to obtain continuing education credits. Please go to this website to answer questions related to this presentation. And with that, I think that's the end. Okay, so I guess it's time to answer questions. Okay. Okay, so the first question I have is, at which stage of pancreatic tumorigenesis do you see upregulation of RalA and RalB? And also at I'm sorry, at PANN2s, PANN3s, pancreatic cancer. Okay, so that's actually a great question. Okay, so as it relates to RalA and RalB, so I've only looked at pancreatic cancer cell lines and tissues. I have not looked at PANNs, and so I actually do not know. Do not know off the top of my head where it was. What's the earliest you see the activation of RalA and RalB? I'm not exactly sure. So I do know, for example, that just although I don't know the answer specifically, what I will say to add to this conversation, K-Ras, we do find K-Ras mutations in PANNs. So for example, studies have been done on autopsy on autopsies, and when people look at doctors look at the pancreas, they see these PANNs, and they find K-Ras mutations in these PANNs. And so, I would guess that because you see the K-Ras mutations, you would potentially see some upregulation of RalA and or RalB in these stages of these earlier stages. I'm sorry, forgive me for those that may not know. PANNs refers to precursor lesions that are believed to go on potentially to form pancreatic cancer. So PANN stands for pancreatic intraepithelial pancreatic intraepithelial neoplasia. That's what the PANN stands for, and that's PANN1, PANN2, and PANN3. And it's believed that the PANN3s are the lesions that are going to go on to potentially become pancreatic cancer. So again, as it relates to your question about upregulation of RalA and RalB, not exactly sure how early you see them, but you do see K-Ras mutations in those early PANN lesions, and so that would make me feel that you would probably see some upregulation in those as well. Question number two, targeting one RAS might lead to proliferation of other types of RAS. Targeting one RAS might lead to proliferation of other types of RAS. That's definitely a possibility. I totally agree. So I dare not say that K-Ras. So although K-Ras is most prevalent in many of the cancers, I won't say that K-Ras, is always acting alone. I totally agree. There may be definitely some collaboration between the RAS, different RAS proteins. Which proteins are intermediaries between K-Ras and PIM, if any? Otherwise, what would be the route that K-Ras would be regulating PIM proteins? Now, that's a great question. That's a very good question, and I'm really trying to figure that out. So, again, PIMs are downstream of the JAK-STAT pathway. Stats have, there have been many papers that have shown stats playing a significant role in pancreatic tumorigenicity. There's definitely papers that talk about K-Ras and stats, and so my hypothesis is I believe stats are playing a role, but I don't necessarily know exactly all the players between K-Ras, stats, and PIMs. I don't know. Now I know there was a paper that focused on nasopharyngeal carcinomas, and I think they looked at PIM1, I think, and they looked at EGFR. So I believe EGFR definitely may be involved in this process as well. But that's a great question, and I'm hoping to spend time figuring that out. What the players between K-Ras and PIM. Great question. Okay, another question. Lately, it has been demonstrated that many oncogenes control inflammation-related pathways. Did you have a chance to check the tumor microenvironment or inflammatory pathways in your downregulated K-Ras in vivo model? Okay, so let me read that again just to make sure everybody heard me. So lately, it has been demonstrated that many oncogenes control inflammation-related pathways. did you have a chance to check the tumor microenvironment or inflammatory pathways in your downregulated K-Ras in vivo model? So I did not get a chance to check. Inflammation, but I really do believe that it's playing a role as it relates to pancreatic cancer. And so definitely something that should be investigated down the road. And I think that's all the questions I had. Thank you for the questions. Thank you for your attention, and I hope you enjoyed it.